Hello everyone, grab your pen and paper and let's talk about type 3 hypersensitivity, the details. First of all, let's understand how this happens, how these arise, and then we will get into the details of how the cells actually die and why they die in the first place. Well, first of all, we've got an antigen presenting cell, in this case the plasma cells. They are going to be presenting the antigens. Let's recall that these antigens are soluble. Now, they are going to be recognizing these antigens and present them on their surface via the MHC class II. Then, who is going to be recognizing them? Essentially, the T helper cells via the CD40 receptor that we've all come to know by now. In this case, the antigens that are present on the MHC class II of the plasma cells, or the antigen presenting cells in this scenario, are autoantigens, so they are antigens from our own body. Now, this is going to be very prominent in people that have a central tolerance that is not really functional, or it is due to their genetic heredity or any other problems that may arise. So it's very important to understand that there's a problem that is happening in our central tolerance, which is leading us to this scenario. If we have a central tolerance system that is good enough, we're not going to be getting these type of hypersensitivities. Now, after the presentation of the antigens onto the TCR-CD3 on the T helper cell, we're going to be having the CD40 receptor that is present on the plasma cell is going to be binding to its ligand, the CD40L, which stands for the CD40 ligand, on the T helper cell to release cytokines. Why are these cytokines important? It's because they are going to be signaling to the plasma cells to produce IgGs, the antibodies, IgGs instead of IgMs. Now, let's recall that those IgGs are going to be binding to soluble antigens, forming the immune complexes that were the antigen-antibody complexes. Now, we have understood now that we are going to be having these antibodies that are going to be binding on these antigens, forming immune complexes. Where is the problem? Why is this such a big deal that it's going to be causing us problems like lupus? Essentially, these immune complexes are going to be forming essentially, these immune complexes are going to be formed from soluble antigens and antibodies, and they will not be able to get eliminated by macrophages. Recall that the macrophages are the white blood cells, or the cells that are present inside our body that are responsible for the removal of the debris. And essentially, when they are not going to be able to remove these immune complexes, they're going to be present in a very large number of cells. Now, why is this also important? It's because since they're not immunogenic enough, these macrophages will not be able to capture them, and they will ultimately deposit at the basement membrane of our cells. Now, it's worth noting here that they deposit due to the opposite charges. Essentially, they have a different charge to the basement membrane, and since opposites attract, they are going to be binding to the basement membrane. Next, what happens? Why is there death of the cells, and why does it cause diseases like lupus? It's because the C1 protein is going to be binding to the antibody that is bound to the antigen, and this is going to be leading to the cleavage of the different complement proteins, namely the C1 to the C9. Now, most notably the C3A, the C4A, and the C5A, as we all know, are anaphylatoxins, and they are going to be triggering an increase in vascular permeability, ultimately causing edema. And also, as we all know, edema is essentially the swelling of a certain part of our body due to, well, the excess presence of the water. And now also, these three proteins are chemokines, which means they can recruit other immune cells to the site of infection. Now, why is this important? Essentially, it is important because these cells that are going to be immune cells that are going to be called upon by these chemokines are going to be neutrophils, and these neutrophils are going to try to phagocyte these immune complexes, and they're also going to fail like the macrophages. Now, when they fail, they are going to be degranulating. They're going to be releasing the granules that are present within them, and these granules that are present within them, now this is such a complicated but such a fun and interesting topic, by the way. So upon degranulating of these neutrophils, they're going to be releasing some reactive oxygen species and lysosomal enzymes. Now, as we all know, reactive oxygen species can be one of the most devastating things that can happen in our body, and this is going to be damaging our vessels by necrosis and leading to vasculitis. So what is dangerous here is that upon necrosis, the DNA and the nucleoproteins that are present within these cells, they're going to be released. And since they are going to be released, the IgGs that were previously released by the plasma cells are now going to be binding to them, therefore forming even more immune complexes and also amplifying the effect of this type of hypersensitivity. So as we all know, and what we can conclude here is the fact that these immune complexes, or this type 3 hypersensitivity, is going to be progressive. It is going to be starting at a point where some cells are going to be releasing these immune complexes or forming these immune complexes, and later these are going to get amplified as more cells are going to be dying and more DNA and nucleoproteins are available for these antibodies to bind to. Now, the levels of the complement proteins present in the blood are going to be allowing us to determine at which state of the disease we're in. This goes hand in hand with our point that we mentioned earlier, where we were mentioning that this is going to be amplified. So the more complement proteins, that means this has been present for a longer time. Now, emphasis should be put on the fact that hypersensitivity is not exclusive to blood vessels and can also be found in the kidneys, causing glomerular nephritis, where the blood is filtered, by the way. We have mentioned that also in a previous video, and joints. And this can cause arthritis, where the plasma is filtered to make synovial fluid. Now, as a conclusion, let's recall everything that we mentioned. First of all, the IgG antibodies, after they are produced or they are subjected to get produced by the plasma cells, it is going to be forming immune complexes with the soluble antigens, which in this case, specifically in this case, are going to be DNA and nucleoproteins. So these IgG are going to be making these immune complexes, and they're going to be depositing on the tissue, and this is going to be attracting the complement proteins, namely the C1 to the C9. Now, also, these immune complexes are going to be, obviously, they're going to be releasing some cytokines. They're also going to be releasing some chemokines, and these chemokines are going to be calling for neutrophils that are going to be releasing reactive oxygen species and lysosomal enzymes in order to destroy these cells. Upon the destruction by necrosis, these are going to be releasing these some DNA and nucleoproteins, and therefore amplifying the process over and over and over again. Thank you for watching. This has been Ali from B Biology.